camera off. That's easier.
Great. All right. Hi, everyone. I'm Kelly MacKinnon, and I'm presenting to you my confirmation of candidature in Silico temporal and spatiotemporal modelling of radiotherapy and immune checkpoint inhibitors in a mesothelioma mouse model. Just quickly going over conflicts of interest. All ethics have been approved by the university and the Harry Perkins Institute. Experiments were in accordance with Australian Code for the Care of Use of Animals for Scientific Purposes, and funding is from the Cancer Australia Priority Driven Collaborative Cancer Research Scheme grant and the Westpac Future Leaders Scholarship Research Fund. So quick, a little background on mesothelioma. So this is Wittenoom in Western Australia. This image depicts an asbestos shovelling competition performed by the miners. Australia has a long history of asbestos usage in mining, and the mine in Wittenoom was only closed in the middle 1960s, and a complete ban of the import and use of asbestos products only occurred in 2005. Mining of asbestos continued in the USA until 2002, and countries such as Russia, Kazakhstan, and China still continue the practice. So one of the key outcomes of asbestos exposure is mesothelioma, where asbestos fibres become lodged in the mesothelium, causing cellular damage leading to cancer. The most common form of mesothelioma occurs in the pleura, the lining of the lungs and chest cavity. This carcinogenic process has a latency period of 20 to 40 years post-exposure. So despite mining bans and reduced asbestos use today, the ramifications of asbestos will be felt for decades. Though mesothelioma is only responsible for 0.5% of Australian cancer cases per annum, it is responsible for triple that in fatalities. Current treatments involve combinations of surgical resection, chemotherapy, targeted agents such as anti-angiogenics, immunotherapy, and radiotherapy. But no one therapy or therapy combination is ideal, and choice of treatment depends on a myriad of factors, including patient health and disease progression. Total remission is rare in meso cases, and treatments are aimed at prolonging life and palliation. Its 6.4% 5-year survival rate is close to half that of pancreatic cancer, giving it one of the worst outcomes of any cancer. And unlike many other kinds of tumours, the survival rate has not appreciably improved over the last 30 years, making it an ideal focus for cutting-edge cancer treatments. So though radiotherapy isn't new, it's still cutting edge. Our understanding is constantly evolving. In recent years, the effect of radiotherapy on the tumour microenvironment, or TME, has become a source of interest. One of Weinberg and Hanahan's hallmarks of cancer is the induction of vasculature or angiogenesis. Vascular growth is normally dormant post-embryogenesis, but during tumour progression, pro-angiogenic factors are chronically activated and anti-angiogenic factors are downregulated. This results in aberrant vasculature with convoluted and excessive branching, distorted and enlarged vessels, and absent or detached perivascular cells, erratic blood flow, and leakiness. This in turn leads to poor perfusion of the tumour and an increase in hypoxia. Poor perfusion means that systemic cancer treatments such as chemotherapy and immunotherapy cannot infiltrate into the tumour bulk and also hampers the infiltration of cytotoxic immune cells. Hypoxia leads to cell cycle arrest, leading to chemo and radioresistance, and hypoxic cells are more likely to metastasize. So fractionated radiotherapy has the ability to normalise the tumour vasculature. This upper image reflects findings in multiple preclinical studies using smaller fractions delivered over time, for example, 10 daily fractions of 2 gray. And this dose and fractionation schedule resulted in vascular normalisation. This functional vasculature, now that it's been normalised, has transiently enhanced blood perfusion and reduction in intratumoral hypoxia. So vascular structures are composed of 2 parts. They have their internal lining of endothelial cells and the external perivascular cells that provide essential stability and structure. So with this radiotherapy fractionation, there is also an increase in pericyte coverage, which is a type of perivascular cell, which is associated with improved blood flow, improved drug delivery, and better infiltration of anti-tumour immune cells. But these effects are time-dependent, with a variety of studies showing that this occurs around 1 to 3 weeks post-irradiation. The vascular normalisation effects also appear to be dependent on both dose and tumour model, with the same fractionation schedule having a different impact on perfusion and oxidation for different tumour types. However, if too much dose is delivered in a given fraction, it leads to RT-induced vascular destruction. This occurs at doses in excess of 10 gray and is most prominent in smaller vasculature. Within 24 hours of this high dose, endothelial cell apoptosis occurs, leading to an increase in vascular permeability and a related temporary increase in perfusion. Within weeks, however, vascular perfusion is greatly reduced as vascular structures die and become occluded, resulting in hypoxia. Pericyte coverage also reduces as they potentially undergo atrophy in response to vascular damage. This hypoxic, low perfusion environment continues or even worsens the response of tumour to further local and systemic treatments. So as well as vascular dysfunction, another core impedance of effective cancer treatment is the tumour immune landscape. Though the immune system is initially geared towards tumour suppression, over time the cancer modulates the immune system in a process called immunoediting, ultimately leading to immune escape and uncontrolled growth. The tumour cells populations present in this escape tumour will have poor immunogenicity, presenting fewer antigens, which is required for the recruitment of tumour-killing immune cells, and also promotes the recruitment of immunosuppressive cells. These tumours are then resistant to further attacks from the immune system. If we take the previous scenario where the vascular normalization has been occurred through radiotherapy, radiotherapy still has a contradictory but simultaneous effect on the immune landscape of the tumour. Irradiated cells tend to die immunogenically through apoptosis, and they release pro-inflammatory cytokines, chemokines, and other danger signals. This increases the amount of antigens available to be presented to tumour-killing T cells. Tumour cell death by irradiation also promotes the presentation of antigens to T cells by the dendritic cells. Coupled with the increased perfusion from the normalised vasculature, the recruited immune effectors are able to more effectively infiltrate the tumour and attack. This increased immune activity can occur in areas remote from the irradiation in the so-called abscopal effect, where tumours outside the treatment site also shrink in response to immune attack. Despite its promise for treating metastatic lesions, this abscopal effect is not commonly observed in clinic from radiotherapy alone. So while this upregulation occurs, we also have a downregulatory effect. Though the situation is, of course, a bit more complicated than what I'm describing here, this is just a bit of a general overview for what is part of my project. So firstly, tumour-killing T cells are among the most radiosensitive of all immune cells. A study in healthy humans found that more than 25% of T cells were killed at doses above 0.5 gray. As well as killing immune effectors within the tumour volume, radiation also recruits immune-suppressing cells, important in regulating the immune response, such as myeloid-derived suppression cells and regulatory T cells. Irradiation also affects protein expressed on the surface of cancer cells. One of the key things is the expression of the PD-L1 checkpoint, which is upregulated across many cancer types, with this change being most notable in normoxic versus hypoxic conditions. The effect of cancer cells expressing more of this protein is that they can identify as self to the immune system, nullifying the cytotoxic T cells and making it easier for the tumour to grow unrestricted. So a little more on these immune checkpoint inhibitors. They're actually themselves an emerging target for cancer treatment. Two of the key checkpoints in preclinical and clinical research are the CTLA-4 and PD-1, PD-L1 axes. CTLA-4 and PD-1 are exhibited most commonly on T cells and PD-L1 on tumour cells. The binding of CTLA-4 to a ligand expressed on an antigen-presenting cell, such as a dendritic cell, leads to an inhibition of the T cell response, reducing T cell proliferation and leading to systemic immune suppression. When the immune system is engaged in a long battle, such as when it is attempting to fight cancer, the overexpression of PD-1 on T cells leads to them becoming exhausted where their functions are inactivated. This means that the tumour only induces a weak immune response and manages to avoid elimination by the immune system and continues the process of carcinogenesis. A way to prevent this is with immune checkpoint inhibitors, or ICIs. These are systemic treatments that block the checkpoint proteins from binding to the inhibitory partner proteins. This blocking can target the T cells themselves, and as in the case of anti-CTLA-4 and anti-PD-1 inhibitors, or can work on the tumour side of the interaction, such as with anti-PD-L1, as you can see in the diagram here. This treatment can have a phenomenal curative effect on some patients, particularly in patients suffering from refractory lung cancer or malignant melanoma. The melanoma is the case of the patient pictured here that received ipilimumab, which is an anti-CTLA-4 treatment. And you can see most notably the lesion on the side of the patient's head has significantly reduced. However, reliable efficacy in the clinical setting is lacking and incidences of serious immune-related adverse effects are high. The addition of ICI to RT means that T cells can have their brakes kind of taken off in the form of the immunotherapy while having a better idea of where to go and better ways to get there from the radiotherapy. Immuno-RT combinations show great promise in clinical trials treating colorectal carcinoma, non-small cell lung cancer, locally advanced and metastatic, melanoma and renal cell carcinoma, with many clinical trials in these and other cancers still ongoing. However, key information on dosage, fractionation and scheduling are unknown, and these parameters must be better understood in order to increase therapeutic efficacy. And that brings us to my project, the in silico temporal and spatiotemporal modelling of radiotherapy and immune checkpoint inhibitors in a mesothelioma mouse model. My primary goal is to optimise radiotherapy and immuno-RT treatments within our experimental context using computational methods. The purpose of this project is to characterise the effects of RT and immuno-RT treatment in a preclinical mesothelioma mouse model, then use the model to interpolate the optimal treatment conditions for complete tumour cure. In this way, the time and resources required for preclinical exploration of treatment conditions can be minimised without compromising on quality and safety as the treatment moves into the clinical trial space. So first, a literature review. Of interest in this review were both the computational modelling of immuno-RT treatments and the modelling of the immunological effects of radiotherapy as a monotherapy on the tumour microenvironment. The goal was to understand previous modelling approaches in order to determine the methodology for this study. In order to do this, broad conditionals had to be used to capture all available studies in English, as there is an inconsistency in the keywords and language used to describe this modelling technique. After filtering through over 1,000 papers, 12 relevant studies were included in the review. The key factors of interest for these models were their methods rather than their outcomes, as most of the models are based upon small cohorts of data, do not quantitatively assess their goodness of fit to the model training data or assess the model's predictive ability. All models consist of partial differential equations describing changes in cellular populations, typically varying populations of cancer cells and immune cells. Of the 12 studies found, 5 were RT monotherapy models and 7 were immuno-RT models. Most models of RT used photon external beam radiotherapy, but 2, both by the same authors in the USA, used proton external beam radiotherapy. The most commonly modelled immune checkpoint inhibitor was anti-PD-L1, and one study chose not to investigate specific ICI, but remained ICI agnostic. Most models explored treatment effects in mice, but one study chose to keep the model patient type agnostic, and a handful of them did look in human subjects. The agnosticism was most notable in the cancer type, where many models chose not to investigate a specific cancer, but instead made it open to any cancer type for wider applicability. Just over half the models explored the effect of different fractionation schedules on treatment outcomes for either the radiotherapy or immuno-RT, and 2/3 of the immuno-RT models looked at the effect of the relative timing of immuno-RT therapy on treatment outcomes. Most of the models were purely temporal, meaning that they looked at the overall change in the tumour cell population over time, but one model incorporated spatial information modelling individual cellular interactions in space and time. For the modelling approach, mathematical models did not use data as the foundation to forming a model, whereas data-driven models made use of specific experimental data. Some models were extensions of already existing models, either making this addition purely mathematical or based upon additional data. Most models were relatively simple with 15 or less parameters, though one had over 30, in fact had over 70 parameters and was far more complex. In some cases, inter-animal variation or inter-patient variation was considered as a parameter within the model, but in many they were looking at population-wide changes in treatment effects. There were few models that actually investigated the fit quality quantitatively, though this couldn't be investigated at all for the mathematical models that weren't built upon data. The predictive quality was only investigated in one model. Most of them chose not to see if the treatment model could predict outcomes in external data sets. And then the stability and sensitivity of the models were analysed in most cases, where stability looks at areas of the model where certain parameter values will lead to unstable growth or lead to tumour suppression, and sensitivity looks at small changes in the parameters to determine which parameters are either most important to the model or perhaps need to be determined to a higher degree of accuracy and specificity to ensure that they don't throw out the model results. So in terms of the literature versus this current project, the model type we will be looking at will be both RT monotherapy and an immuno-RT model. We'll be using photon EBRT and both anti-PD-1 and anti-PD-L1, anti-PD-1 and anti-CTLA4 ICI type. The subjects will be mice and looking specifically at murine mesothelioma. We will be investigating the effects of both fractionation and immuno-RT scheduling to get the best understanding of the conditions that will most affect the optimization of treatment. We'll be doing both temporal and spatiotemporal models, where the temporal model will be data-driven in both RT and immuno-RT cases. Will be data-driven in the RT cases, but in the immuno-RT cases, this will be likely just a mathematical extension to add in the slightly less complicated effects of the immuno treatments. The goal will be to keep the number of parameters low, to keep the simplicity of the model, to make it a bit more applicable to clinical and preclinical research. We will also be considering inter-animal variation to try to understand what potentially tumour immune landscapes pre-treatment or any other factors could lead to treatment efficacy. We will also make sure that we investigate the fit and the predictive quality quantitatively, and we will also analyse stability and sensitivity of the model in order to get the greatest insight possible. So the three aims for my project. The first one is the in silico modelling of RT monotreatment, with the two sub-aims of doing a temporal model and a spatiotemporal model, where the spatiotemporal model will include the movement of the cells as well as just the gross tumour dynamics that are occurring. The aim 2 is essentially identical to aim 1, but for the immuno-RT synergistic treatment. And then aim 3 will be the external testing of the in silico RT monotreatment model predictions by actually generating new data to feed back into the model to test its prediction ability. So the method for aim 1 and 2, as mentioned, virtually identical, so I've coalesced them together. So initially, the first thing to do is a literature review to find patterns in modelling methods in order to shortlist model training and testing methods. At the same time as that, it's actually getting access to the data that's already being generated by Sanat, Keem and Rebecca Delonzo, my fellow PhD students, and figuring out how to convert the data into a usable format and extracting the information. Once the ideal model formats have been determined and the data extracted, the data will be split into testing and training sets so that the model can be tested to see whether it works within its own cohort of information that has been used to train it. And then ranges will be selected for the unknown parameters based upon common values in literature. The models will be tested upon the reserved data that wasn't used to train the parameters, and then the best model or models will be selected from this. So in terms of data, we have a lot to choose from, 5 different experiments, where the blue experiments are for the radiotherapy model and the green experiments are for the immuno-RT model. So experiment 1 is testing radiotherapy fractionation schedules and their impact on tumour vasculature and the immune landscape. Experiment 2 is determining the optimal immunotherapy and radiotherapy scheduling for tumour cure using results from experiment 1. Third is investigating the transient effects of optimal RT fractionation schedules on tumour vasculature and the immune landscape. Number 4 is investigating the effects of optimal immuno-RT scheduling from experiment 2 on larger tumours, as there's the understanding that the size of the tumour when the treatment is administered can make an effect on outcomes. And fifth, we'll be investigating the effects of optimal immuno-RT scheduling on a different mesothelioma subtype in a different mouse model. So overall, that leads to a fair bit of data. So we have essentially the two different kinds of imaging. We've got ultrasound and immunohistochemistry. So the ultrasound is conducted in vivo while the mouse is still alive, and the photoacoustic imaging lets us see oxygenation information, while the power Doppler imaging lets us see perfusion information. And then the immunohistochemistry is performed once the tumour has been resected, and then the tumour slices are stained for different proteins that correlate to different cells and different functionality. So we have the CD31 is a vascular stain, which gives us information of the location of vasculature. CD3, CD8, CD4, and Tregs are all different kinds of T cells with either cytotoxic or regulatory suppressive phenotypes. The peri are pericytes, which are an indicator of vascular health, as mentioned previously. Lect or lectin is a perfusion marker, so we can see how much blood is actually getting into vasculature. PIM is for pimonidazole, which is a hypoxia marker, and collagen is another indication of vascular health. So between these 5 different experiments, with the purple ones that are still in process or to be commenced, we have a fair bit of data to go off. The yellow pimonidazole in the first row sadly didn't take too well, but it has been perfected for experiment 3 and we'll have access to it then. So for the AIM3 method, this is for the actual designing of an experiment, an animal experiment, to test to see if the optimization, the optimized treatment scenarios as given by the RT monotherapy treatment model are accurate, and then also to generate more data so that we can put it back into the model and further see how well the model predicted the outcomes. So we need to obtain the optimal model conditions from the model itself, and that'll also allow us to quantify the treatment effect size, which will let us determine what size cohort of mice are required. And so then applying for ethics and enlisting either an honours student or an RA to perform the bulk of the experiment, we'll need to perform the experiment and generate the data in the same way that the previous experiments have been gathered. So we're lucky enough to have treatment protocols and imaging protocols already in place. We will then perform the same things as the previous modelling methods in order to test the model and quantify its accuracy to see how well the model building has actually correlated with preclinical outcomes. In terms of costs, the costs to the university are quite minimal, the joys of a computational project at this point, and the mouse part of the modelling will likely be incorporated into some other experimentation that will already be occurring. And in terms of additional expenses, I'm lucky enough to have a Westpac Future Leaders Scholarship that has helped to supplement my income, but also my funding from UWA, so that I'm able to invest in more development programs and further training. So looking at a progress timeline, I'm not going to dwell on this because I've had some ongoing health issues for a while, and though this was submitted with my annual report, I've since had to take 2 months off and reevaluate some of my timelines since this. So instead, I'll go into the actual progress that has been made. So in terms of further study, I've completed a statistics unit multi-level linear modelling at UWA. And on Coursera, using Westpac funds, I've completed a variety of biological modelling courses. These courses have enabled me to understand the current literature on in silico treatment modelling and develop a detailed plan for assembling, fitting and testing a biological treatment model. This has been crucial because my physics background has not been very heavy in biology or statistics, so being able to do these units has been very valuable. In further training, I have now completed all formal animal training and induction processes. I've been signed off on these experimental skills by the supervising vet at the Harry Perkins Institute, so I can aid with animal experiments and have an understanding of the procedures necessary for planning an experiment for AIM3. having knowledge of how the data are generated also leads to a more relevant and accurate mathematical model. And so I was very keen to be able to get in at this part of the process so I'd understand the context around the data that I will be fitting within the model. In terms of submissions, I've submitted my research proposal and I've been added to the current ethics that we were working with. I've also submitted my annual report and my literature review is in progress. I'm also a co-author on 2 publications, with Rebecca Delonzo and Sanat Keem being each a lead author, and Sanat Keem has just had another paper accepted that I'm also a co-author on. In terms of data extraction, so for the immunohistochemistry images, there's a bit of complexity with conversion of proprietary image types, but we're now at a point where I can convert it to a NumPy array, which makes analysis much easier. I'm also working with a software developer friend to create an open source software for vascular image analysis of these immunohistochemistry images to semi-automate the process. I can't go into too much detail because we've been encouraged to apply for a patent and sort out the IP, but the goal is for that to be able to save a lot of time and effort for researchers and investigators. And we're quite excited with how it's going so far. I've also determined the radiosensitivity of the mesothelioma cells, the AB1HA cells. It was determined using MATLAB and clonogenic survival analysis data, but now that I have a better understanding of Python at this point in my project, I'm going to revisit that measurement with the methods that I'll be using for the rest of the project. So in terms of setbacks, initially there were some financial setbacks. Research funds were not released immediately by the university, which prevented purchase of core equipment. It took about 5 months for the funds to be released after I started my PhD. So this did take a fair bit of time to sort out and did set progress back a bit. In terms of image processing, unlike in clinical image processing, where images are most commonly in the DICOM format, preclinical imaging is dominated by niche proprietary formats where providers provide their own, often expensive and restrictive licenses to process the images using their own software. Typical command line or Python methods don't always work to convert some of these image types, and finding a way to unify image processing for multiple image modalities has proven quite complex. Also, as mentioned, in terms of health, I've had to have, I think, collectively a bit over 4 months off since I started the PhD. So this has very much set my timeline back as well. In terms of solutions, though, for the financial issues, I worked with UWA to overhaul the payment method of the Westpac scholarship. So now current and future students have easy access to their funds and are not held up in their work. So that was a lot of time and effort, but that's been a really great outcome. For the image processing issues, as mentioned, working with a software developer makes things a lot easier. So he's already managed to solve problems that no forum had an answer to. So we're really looking forward to releasing those online to make other people's lives easier. Health problems not quite as easy of a solve, but we'll see how that goes. I'm constantly optimistic and we'll see how that goes over the next few months. The next tasks are to complete literature review and prepare it for publication, to complete the IHC image processing pipeline and ready the software for release, and also to complete the 3D Doppler image processing pipeline. Lastly, for the extra activities that I've been able to do during the PhD, I've had the joy of being able to take part in a leadership development program through the Westpac Future Leaders Scholarship, where they flew us over to Sydney and put us through a very rigorous training and development program. It was very enjoyable and very exhausting, but I'm very much looking forward to the next one in September. I'm also one of the founding members of Ease ADHD, which is an ADHD mentoring program we're putting together for people in their postgraduate studies and towards the end of their undergraduate studies who got a diagnosis later in life and are wanting to try to figure out some techniques that will be able to help them in their studies and in their everyday life. And lastly, I've been working with the Centre for Advanced Technology and Cancer Research, helping to establish that with some of the wonderful people that are here today and some other wonderful people that aren't. This is an extremely exciting project that a lot of us are looking forward to, the idea of creating a cancer centre for excellence within our own group here. So acknowledgements. Thank you to my supervisors for your absolute patience and just for being absolutely fantastic. To Rebecca and Sanat for your fantastic help and for being such excellent fellow researchers. To Byron for being a software whiz. To the Medical Physics Machine Learning Cluster for their excellent advice for coding questions. And to the Westpac Scholars Group for their ongoing support. Thank you, everyone. Are there any questions?
Great. Thanks, Kelly. Excellent. Any questions from Kelly?
Yes, Brandon.
Sorry, that was a mistake. I was meant to do the clapping version, but I just mistook for hand rise. Don't worry, there's no questions. But
Yeah,
good
that's right.
presentation though.
Thank you. I'll take it as a high five then.
That's great. It was a very clear presentation.
Oh, good. I'm glad.
I got a question.
Yes.
You can get away with that easily.
No, I was hoping there'd be at least one. I'd feel like I got off too easily otherwise.
Absolutely. I guess so you put your aims down, which are really about forming the models, right?
Yes.
So you've got the model now, and you've validated it, so you can model these mouse mesothelioma tumours and their response. What comes next?
So that would be most encompassed under AIM3. So AIM3 is actually taking what the particularly the RT monotherapy model indicates as being the optimal treatment conditions in terms of fractionating of dose, immunotherapy, radiotherapy scheduling, and ICI dosage, if that's something that I have the ability to look into, and then designing that preclinical experiment based upon those optimal conditions to see if the model does actually give an idea of what optimal should be, because the point of the project is to be able to expedite that preclinical research. And currently, none of the models out there have really been validated in such a rigorous way that they'd be able to be used in that kind of forward planning kind of method. In fact, only one is by the group at Harvard, who have actually been able to identify potential cohort sizes and treatment effects to be able to look at future research. So the ultimate goal is to make this process quicker, which I suppose the test of that will really be in the AIM3 to see if it does fulfill that purpose. But so much of the import is put on actually building the model, because selecting each of those little parameters and the modeling method and how to process the data without spending a lot of time on that part of the process, there's almost no point trying to do anything with the model, because many of the papers I've looked at, they've kind of made all these grand statements of how many days treatments should have between them and what size fractions there should be, but they haven't actually used that much in the way of data or rigorously validated their model, or they're speaking outside what their data is actually allowed to tell them. So the key foci for AIM 1 and 2 is actually to build something good, and then AIM 3 is to test to see, is it actually any good?
So I guess the extension to that is that'll help us predict response of mesothelioma that we put into mice. Well, how does that then help us clinically?
So part of it, the first part would be the help in clinical is being able to get through preclinical quickly and accurately, because either way it'll be validated in experiment. So if preclinical is able to be expedited, then that helps clinical ultimately. But also this model is a bit more optimized for preclinical research as well, because it's using the data that we're gathering is a mix of in vivo, where we are getting the images from the tumours, but it's also a lot of tumour cross-sections, which is only something you can have after the tumour's been actually excised. And particularly with mesothelioma and the way that it grows, it doesn't grow in the nice, neat little lump so that it can be cut out of a human patient and then cross-section stained to get an idea of what the internal characteristics are. So the models that are better for actual clinical kind of measurements, they use more in vivo, such as blood measurements while the patients are undergoing treatment, which is actually something I'd like to investigate in AIM-3 to see if the circulating lymphocyte population has an effect or can be a biomarker of how the treatment's going or what their initial levels were to determine if they're going to respond well to treatment. But based upon the data that we currently have, that is more preclinically optimized, but I would like to extend through AIM-3 to gather some more data that would lead it more towards being clinically applicable as well. Gosh, you really see how much you talk when you've got AI transcribing what you're saying while you're saying it. Any other questions?
No questions, seems all good. Thanks very much, Kelly. So could you please stop recording?
There we go.